Hello and welcome back. The topic for the next series of videos that we're going to discuss are going to be cells. So we are going to discuss what makes up a cell, the different components, the functions of those different components, how each of those components interact with one another, what makes up a cell, how you visualize cells. All right, so we've got a lot of topics to cover this chapter. And we're going to start off talking about microscopes, because cells are small. All right, we're talking on the range as far as size. All right, if we're talking a bacterial cell, maybe 1 micrometer. If we're talking a human cell, somewhere around 10 micrometers, although there is a lot of range as far as size of eukaryotic cells in the body. You can have some larger cells on the lines of 50 micrometers, all right? And those numbers probably right now don't mean a gosh darn thing to you, all right? You have no idea what 1 micrometer versus 10 micrometers means. Although hopefully you'll realize that 1 is smaller than 10, okay? So a bacterial cell is going to be smaller than a eukaryotic cell, all right? But to give you some context, all right, the limit of what we can see with the unaided eye is 200 micrometers, all right? So that's, in terms of that, that's a larger number than the 10 or the 50 that I talked about with the eukaryotic cells. And to kind of give you some context for that, what the heck is 200 micrometers? All right, 200 micrometers is about the diameter of human hair. So if you take an individual strand of hair, if I held up a strand of hair, you wouldn't be able to see it with these video conditions. And even if I was at the front of a classroom, maybe the front row would be able to see that hair I was holding, but probably not, all right? But if you hold that up, you can see an individual human hair. That human hair, that's about 200 micrometers, all right? And what I like to kind of give you an idea on cell and cell size, I've got a couple of links. So check Blackboard for the links that I have posted. One of the links that I really like, and this you can just Google and it should come up, you should find it, is called Cells Alive. It's a nice website. They've got some interactive videos for you to see essentially how big something is as far as cell size goes. It also has some nice diagrams of the cellular components, which will be kind of the topic of some later videos. So I've got a number of links for you to check out that can kind of help with you figuring out what that size means. Because in lecture, it's a lot harder for me to communicate the size. If you're taking a lab, it's a little bit easier because you actually get to see the cells. You actually get to look through a microscope, all right? And I will try and post also some links of some virtual microscopes in case you're not taking the lab course this semester, to kind of give you an idea how the different sizes of different cells kind of plays out. Prokaryotic versus eukaryotic. All right, but the important part there, cells are small. So in order to be able to visualize cells, we need microscopes, okay? The microscope magnifies the image. So if you think about a magnifying glass, all right, you can use that. That makes things look bigger if you look through a magnifying glass. That's essentially what the microscopes do. And with the microscope magnification, all right, you're seeing something larger than it is in real live life. And the way we kind of express the fact that you are seeing something at not its usual size is to put some number followed by an X. All right? That X tells you that you have some magnification associated with it. In this case, 20x would tell you that you are seeing something 20 times bigger than it is normally. All right? When you look through a typical light microscope, cells, you're going to be able to visualize anywhere from, 100x to 1000x magnification. All right? Eukaryotic cells are going to be easier to see at this level. The 1000x magnification, that's what you're going to need to be able to visualize prokaryotic cells. All right? Because prokaryotic cells fall into this category. They're somewhere around a micrometer. Eukaryotic cells, somewhere around 10 micrometers. All right? But the way we figure out magnification is based on the lens associated with the microscope. And with the microscope lens, it's called the objective lens. And typically you'll see numbers on an objective lens, 4x, 10x, 40x, 100x, if you're taking the associated lab here. Those are the common objective lenses that we have. But we also have, I'm going to put it in a different color, an ocular lens. And that ocular lens is going to be the lens that you look through to be able to see the cells. Okay? The ocular lens, for most scopes, is usually about 10x. Okay? So what we have to do in order to determine how much magnification we are visualizing a particular sample at is we need to take the objective lens number and multiply it by the ocular lens. So in our case, our ocular lens is 10x, okay? And we're going to multiply that 10x by, let's say, 40. All right? And 40 times 10, usually in class I stop and ask you what this equals. In videos I can't do that. 40 times 10, that's going to give you a total magnification of 400x. So when you see total magnification, if I ask you to tell me what the total magnification of something is, you need to know the objective lens magnification, you need to know the ocular lens magnification. You multiply those two numbers, total magnification would be 400x. Okay? And most cells, eukaryotic cells, you'll be able to see a good amount of detail at 400x. You'll start to see it at some of the detail at 100x. You just get a little more detail at 400. Prokaryotic cells, you need to go up to 1000x total magnification. And I'm going to put these in green because the numbers I'm talking about here are total magnification. Okay? So that's kind of some simple math. In terms of this class, that's about as complicated as the math is going to get for this class. So I expect you to be able to tell me what total magnification is associated with some number that I give you. And what you'll see, if you go to the Cells Alive site or the virtual microscope, you'll see differences in what you can see at various magnifications. All right? You're going to be able to see more detail at a higher magnification, so 400x, 1000x, but you're going to see a smaller field of view. So when you increase the magnification, your field of view in the microscope gets smaller. So you're concentrating on a smaller and smaller piece of whatever tissue or sample that you're looking at. That's just kind of one of the characteristics associated with microscopes. All right? With microscopes, there are going to be two types of microscopes that I'm going to talk about. One is going to be the light microscope. And if you're taking 1492 lab, this is going to be the type of microscope that you're using in lab. It's a light microscope. The other type of microscope is an electron microscope. We don't have an electron microscope here on campus. Electron microscopes, cost-wise, cost a lot more money, okay? Electron microscopes can start anywhere at $100,000 and go up. So that's why we don't have electron microscopes for you to use. Electron microscopes are very delicate pieces of equipment, so you have to undergo a lot of training in order to use an electron microscope. With the electron microscope, we're going to have two types of electron microscopes. We're going to have what's known as an SEM, and we're going to have a TM. Okay? The S stands for scanning electron microscope. The T stands for transmission. And there are going to be some differences in what you see between a scanning and a transmission electron microscope. Just like there'll be some differences you'll see as far as a light microscope. I have some images in the PowerPoint for you to look at on the differences between a light microscope, a scanning electron microscope, and a transmission electron microscope. So you can kind of see the difference as far as what you can see. You get to see a lot more detail associated with the electron microscope because the electron microscope has what's known as greater resolution. All right? And when we're talking about resolution with microscopes, we're talking about the ability to see 2 points as distinct points. All right? So I typically, when I teach face-to-face, I'll draw things on the board, and I'll draw things really close together. And in the video, you can probably still see that there are 2 dots there. All right? But if you walk away from your computer screen and go further away from the computer screen, further away, you may not be able to see those now as 2 distinct dots. And that's what we're talking about with resolution, all right? So typically, you know, and this might be a little easier to do that with, these are 2 dots right next to each other, all right? Resolution gets at being able to see 2 individual points as distinct points. So electron microscopes have greater resolution as compared to light microscopes. However, there are some advantages to a light microscope. A light microscope, you can view either live or dead samples. Okay? So the advantage here with the light microscope is you can visualize something that's alive. The electron microscope, the samples, because of the processing or the process needed to attach the sample to the slide and be able to visualize it using the electron microscope is not compatible with life. So anytime you're looking at an electron microscope image, the sample you're looking at is no longer alive. All right? It's not alive, but you can see a lot more detail. Okay? And hopefully the images I have in the PowerPoint, you can see some of that. The differences between prokaryotic cells and eukaryotic cells, what you can visualize with a light microscope versus an electron microscope. All right? Hopefully that makes sense. I've got a couple more videos to kind of go over not a couple more videos. I think I've got one more video to go over characteristics and what you can see through a microscope. I'm going to go into a little more detail with the light microscope versus the electron microscope. If you have any questions, please let me know.